In this video, we are going to be talking about the cell, its structure and function, and the organelles within the cell. So cells make up all living things on this planet, whether they're unicellular or multicellular, they are going to be made out of cells. Cells can range in size from 10 micrometers to 100 micrometers, and they vary in size, shape, as well as function. So just to elaborate on this point, speaking of human cells, an egg cell and a sperm cell have very different sizes. An egg cell is the largest cell in the body, and a sperm cell is one of the smallest cells in the body. Similarly, we can think of red blood cells and how their shape differs. We can think of a neuron and how its shape is different from a regular cell that we think of. When we think of a cell, we think of a round object, but cells can vary in shape, size, and obviously their function as well. For this video, we're going to focus on an animal cell. So what you see on your screen right now is an animal cell. And it has all of these organelles that we will be talking about one by one, starting with the cytoplasm. So the cytoplasm consists of two things. It consists of the cytosol as well as the organelles. And the cytosol is the gel-like substance that the organelles live in. So between the nucleus and the plasma membrane, which is all of the stuff here, that is the cytoplasm. And again, the cytosol is the gel-like substance, and the organelles that are living in that cytosol, all that together combined is the cytoplasm. The plasma membrane is the membrane that will surround the cell. So if this is the cell and this is the nucleus, this would be the plasma membrane. I'm going to call it PM. Sometimes your teacher might call it the cell membrane. It is the same thing. Plasma membrane, cell membrane, it is the same thing. It is also called a phospholipid bilayer, and it is selectively permeable, which means that it will pick and choose what substances can enter and leave the cell. Why is it called a phospholipid bilayer? So let's break the words down. Bi means two. So it is a layer, or actually it is two layers of phospholipid. So if I zoom in here, you will notice that there is this one layer of phospholipids. Okay, we'll call it layer 1. And then you have the other layer on the bottom. So it's two layers, bilayer of phospholipids. Now let's see where the phospholipid part comes in. So if I take one of these here and show you what it looks like, this is what it looks like. This has a phosphate group attached, the head does. So it has a phosphate head, and these tails are two tails of fatty acids, in other words, lipids. So that's where the word phospholipid comes from the head having a phosphate group and the tails being lipid, phospholipid. The head is hydrophilic, which means that it loves water. And the tails are hydrophobic, which means that they are water hating. They do not like water. They do not like being surrounded by water. And so overall, we say that the plasma membrane is waterproof. It does not allow water to enter through it. But the cell does need water. So the cell has specific pores that are meant for the entry and exit of water. The nucleus is where the DNA lives, and the nucleus is surrounded by a nuclear envelope. In other words, it's also a membrane. Just like the cell itself is surrounded by a membrane, the nucleus is surrounded by the membrane, and we call it the nuclear envelope. That nuclear envelope has pores in it. It has these openings in it. And those openings are present because inside the nucleus, mRNA forms. And so that mRNA has to leave the nucleus, and so those pores allow that to happen. There are also ions that need to enter the nucleus, and so those pores allow them to enter as well. The nucleus has a darker stained region right here, which is called the nucleolus, and the nucleolus makes ribosomes. Ribosomes are an organelle that we're going to talk about soon, and they are responsible for making proteins. So ribosomes, like I said, they make proteins, and they consist of two subunits. So the one in orange right there, that is the large subunit, and this one is the small subunit. So a small and large subunit together will make a ribosome. And ribosomes can exist freely in the cell, or sometimes they will be bound to the endoplasmic reticulum. And when they're bound to the endoplasmic reticulum, we call it the rough endoplasmic reticulum because it gives it a rough appearance under a microscope. So, like I said, ribosomes are involved in making proteins. We will find that in the pancreas, the pancreatic cells will have a high number of ribosomes because pancreatic cells are involved in making digestive enzymes. And an enzyme is a protein. So pancreatic cells are making lots and lots of digestive enzymes or lots of proteins, and so they will obviously have lots of ribosomes so that they can make enzymes. Mitochondria is called the powerhouse of the cell, and the reason why it is called the powerhouse of the cell is because it makes ATP. What is ATP? ATP can be thought of as the battery of the cell. It holds energy within it, and so every time the cell needs to um, conduct a process that needs energy, it will break down a molecule of ATP and release that energy. So the mitochondria is surrounded by two membranes. It's kind of hard to see here unless you focus really good, but there's an outer membrane and then there is an inner membrane. And the inner membrane is folded like that, and those folds are called cristae. And the inside of the mitochondria is called the mitochondrial matrix. Muscle cells contain many mitochondria because muscle cells use a lot of energy. They use a lot of ATP. So they have a lot of mitochondria so that they can keep up with the energy demand. Peroxisomes and lysosomes are next. So peroxisomes and lysosomes are structures that can be a little bit confusing because they look alike and they have similar functions. I want you to think of peroxisomes and lysosomes as the janitorial staff of the cell because they're getting rid of stuff that the cell no longer needs. We will go over the main differences between the two so you can remember and differentiate. So just so I can point out, peroxisomes are right there, the green ones, and then the lysosomes are shown here in pink. So as you can see, they look alike. Peroxisomes are membrane bound sacs, which means that they are also surrounded by a membrane. They detoxify fatty acids, amino acids, and hydrogen peroxide. So hydrogen peroxide is the main thing that they will detoxify. And when I say detoxify, I mean they break it up. Hydrogen peroxide is H2O2, and it is something that is toxic to cells. It is released when the cell is carrying out its different activities. However, having it inside the cell is toxic. So peroxisomes will break hydrogen peroxide into water and oxygen. Now both water and oxygen are harmless to the cell. Peroxisomes contain an enzyme called catalase, and that catalase is responsible for breaking down hydrogen peroxide into the harmless products. Liver cells that carry out detoxification of alcohol, of drugs, of other toxins will have lots and lots of peroxisomes. Now let's move on to lysosomes. Lysosomes are also membrane bound, but the difference between peroxisomes and lysosomes is what they break up. So where peroxisomes were breaking up fatty acids, amino acids, and hydrogen peroxide, lysosomes are going to be breaking up two kinds of things. One, they're going to be breaking down polymers, and two, they're going to be breaking down organelles. Those are the big things that they're breaking down. They do other things as well. So a polymer is a long chain that is made out of monomers. So a polymer will be a chain made out of single units, which are called monomers. Mono means one, poly means many, and mer means unit. So polymer, many units, and monomer, one unit. Polysaccharides, proteins, nucleic acids, and lipids are all examples of polymers. So lysosomes will break those down in a cell. They also break down organelles that are worn out, that are non functioning, that the cell no longer needs. The lysosome will fuse with that organelle and digest it. How does it do that? It has hydrolytic enzymes that are very acidic, and so those will digest those worn out organelles. Another difference between peroxisomes and lysosomes is that lysosomes are not found in plant cells, they're only found in animal cells. Vacuole. So, vacuole, I have an arrow pointing right here at the vacuole. Vacuoles are also membrane bound sacs, but they store stuff, they store material. In animal cells, they will store waste, and in plant cells, they will primarily store water. Endoplasmic reticulum. So, this is a series of interconnected membranous sacs and tubules that is basically continuous with the nucleus. So, right outside the nucleus, you will see the endoplasmic reticulum. And the endoplasmic reticulum comes in two categories, of two kinds: rough ER and smooth ER. So, what's the difference? The rough ER is surrounded by ribosomes. And because it's surrounded by ribosomes, it is involved in, or I should say, carries out protein synthesis. Proteins are made here. The smooth ER, so if you follow this, here is the smooth ER. The smooth ER does not have ribosomes, so I'm going to say no ribosomes. It makes lipids and steroids. Lipids and steroids. For labs and such, you will also, or for labs and such, a good way to remember where the endoplasmic reticulum is that it's going to be right next to the nucleus. Moving on, we have the Golgi body or the Golgi apparatus, and the Golgi apparatus is going to be not right next to the nucleus but close to the nucleus. So if I go back here to our original diagram, the Golgi body is right here. The reason why it is close to the endoplasmic reticulum is because it is receiving stuff, which are proteins and lipids and steroids. It is receiving all of those and taking them in and packaging them and sorting them. So Golgi apparatus modifies, sorts, tags, packages, and distributes lipids and proteins. So in the diagram here, you can see that right here, this vesicle is butting off the rough endoplasmic reticulum and going into the Golgi body, the Golgi apparatus, where it will go through the tubules. And in the tubules, the Golgi will modify, sort, tag what it's receiving, and then ship it off on the trans site. So it'll ship it off, and that package will go wherever it is needed. What do we mean when we say it modifies, sorts, and tags? So as the Golgi is receiving vesicles, they are meant for different parts of the cell. Sometimes they're meant for going outside the cell, for example, hormones. So it'll sort it kind of like the FedEx or the UPS. It will sort to say this package needs to go inside, this package needs to go outside. It also modifies them, so it will perhaps attach a sugar group or a phosphate group to what it is receiving, and then they'll butt off as they're doing here, and then they will go where they are needed. The cytoskeleton of the cell is a skeleton, just like we have our skeleton that helps us move, it helps us maintain our shape. The cytoskeleton does something very similar. It is made out of proteins, not bones like ours is. It helps maintain the cell's shape. It secures organelles in their specific position there, so they're not moving around, sloshing around. It also allows the cytoplasm and vesicles to move within the cell. So vesicles are little Ubers or little taxis within the cell that are taking material where it needs to go. And they don't have like a jet engine to power them. They need help getting to their destination. So the cytoskeleton actually provides tracks for them, much like a railway track. It provides tracks for these vesicles to move on. The cytoskeleton also enables cells within multicellular organisms to move. So, a multicellular organism, an example of it is us. In our body, we have several cells. One of those is white blood cells, and white blood cells actually chase and move around. And so, for that to happen, the cytoskeleton has to move and make that happen. The cytoskeleton is made out of three components: microfilaments, intermediate filaments, and microtubules. We'll start off with microfilaments. So, microfilaments are the narrowest of the three. They are 7 nanometers in diameter. So, right there is about 7 nanometers. They are made out of 2 strands of actin subunits. Each little green circle that you're looking at, that is an actin subunit, and actin is a kind of a protein. So you have 2 strands of actin subunits intertwined, making up a microfilament. They will assemble and disassemble on demand. So whenever the cell needs a microfilament, it will form. When it's no longer needed, it'll disassemble. They are involved in cell division and cell movement. So when our cells divide, they change their shape as they are dividing. So this kind of a shape right here, the cell, something needs to happen so that that shape changes, and those are microfilaments that are moving around, making the cell be able to have that shape, that kind of a shape. They also help in cell movement. So some cells within our body, like the white blood cells here, so this is a white blood cell, and it is chasing after this teeny tiny bacteria here. And our white blood cells will patrol around, move around, and look for pathogens. This particular one is completely changing its shape and moving around and maneuvering, and eventually it'll engulf that bacteria. So again, there's something happening within this white blood cell that enables it to move and maneuver like that, and it is the job of the microfilaments to help it move. Just like our skeleton system helps us move. Well, similar. Cells don't have muscles, we do. Lastly, it will provide shape and rigidity to the cell. So not only is it helping the cell move, it is helping it change its shape, and it is also providing it with rigidity. Next, we have intermediate filaments. These are made out of fibrous proteins that are twisted together. So one of the fibrous proteins that makes up intermediate filaments is keratin. Keratin is a fibrous protein. Every time you have a fibrous protein, it's going to give that particular substance some toughness. They are not involved in cell movement, but they help in maintaining the cell's shape by bearing tension. Remember, they're fibrous proteins, so they're strong. They can bear tension. They can bear compression. They are spread all throughout the cell. So if I zoom in and I find the microfilaments, if I find the intermediate filaments, they're the blue ones that are kind of scaffolded around the edge of the cell. But the truth is, they're all over the cell. They're transpersed all throughout the cell. And so they hold they hold the nucleus, or they anchor the nucleus and the other organelles in place. If we didn't have these microtubules, these fibrous proteins holding the nucleus and the organelles in place, and they would be moving around. Finally, we have microfilaments. And microfilaments are hollow tubules. They are the widest of the three components of the cytoskeleton. They are made out of alpha tubulin and beta tubulin, which are proteins. And they resist compression like the springs of a mattress. So when you sit on a mattress, the springs will take the compression. Microfilaments do the same within the cell. So if it's squished, it won't just compress. It'll bounce back, if I can say that. They also provide tracks for vesicles to move, much like railroad tracks. So, like I mentioned, vesicles need to move around. They don't have a jet engine that powers them, but they have to have tracks that are laid out for them that they can go on. They also pull replicated chromosomes apart. So this happens during cell division, during anaphase actually, when the chromosomes, replicated chromosomes, have to be pulled apart on either end. It is the microtubules that will pull them apart. Flagella, cilia, centrioles are all made out of microtubules. Centrosome. So centrosome refers to this area. So it is the microtubule organizing center found near the nuclei of animal cells. Plant cells don't have centrosomes. And the centrosome is made out of two centrioles that are perpendicular or at right angles to each other. And these centrioles, if you look closely, they have these microtubules in them. And they will replicate the centrosome. The entire centrosome will replicate. It'll make a copy of itself right before cell division. And it'll migrate to either ends of the cell. So you're going to have one here and one here. Those are the centrioles at right angles to each other. And during cell division, it'll release those microtubules that we just talked about, and those microtubules will pull the chromosomes to either side of the cell, eventually giving you two daughter cells.